The kidney is a complex organ. We've all been looking at multiple signaling pathways to try and find a way where we can prevent progression and stop kidney disease.
Right, we're not saying we're just trying to slow it down. We're not saying, just live with it, just manage it. We're saying we want to cure patients, and I think that's a quite admirable goal.
Being able to have people from different sides of research, including the more clinical side, the more laboratory-oriented side, because people will bring different questions to the table. Having those different opinions and different questions really can lead you in new and novel directions with your research.
We have to constantly push the envelope. We have to generate new knowledge, and this is where we find the cure for diseases that we want to treat.
So we've got multiple pieces to the puzzle, but it's basically putting them all together that's going to drive research forward.
Hello, I'm Sue Quaggan, this year's chair of ASN's Award Selection Committee. The American Society of Nephrology and the American Heart Association Kidney and Cardiovascular Disease are proud to present the 2020 Donald W. Seldin Young Investigator Award. Dr. Seldin, a pioneer in our field, helped to establish the subspecialty as well as the American Society of Nephrology. He was the Society's second president. I am delighted to present this year's Donald W. Seldin Award to Dr. Anna Greka, who will present on From Genes to Mechanisms to Medicines, the Arc of Discovery for Kidney Diseases.
It is a great pleasure and a great honor to be with you today to accept the Donald Seldin Award of the Young Investigator Award of the American Society of Nephrology, and to share with you some of our recent work in this area. The title of my talk is Genes to Mechanisms to Medicines, the Arc of Discovery for Kidney Diseases. And I would like to give you two stories that represent some of our work in this area over the last few years. These are my disclosures, especially my financial interest in Goldfinch Bio will be relevant to the first story that we share together today. So a fundamental principle for a lot of the things that we do in my laboratory is really driving this connection between discovering genes, genetics and genomics of human diseases, understanding the molecular disease pathways that these genes mediate, and then ultimately, through that fundamental understanding, being able to develop precision medicines, which to us means matching the right patient with the right treatment at the right time. So how can we accomplish this goal? This is definitely a very lofty goal. One of the stories that I will share with you today has to do with the glomerulus, the filter part of the kidney, and podocyte injury as it relates to focal segmental glomerulosclerosis, a disease that is very prevalent in every part of the world and for which we badly need new treatments. So how can we go about connecting from genes to mechanisms to medicines for something like podocyte injury and FSGS? When I first started in this field, I was very struck by the fact that very prominent scientists in the field, like Andre Shaw and Friedhelm Hildebrand, had essentially discovered a whole series of mutations that, when taken together, resulted in a singular nodal event, and that was the activation of Rac1. There were so many different autosomal dominant as well as recessive mutations that drive FSGS that seem to coalesce on the activation of Rac1, as were in some cases of sporadic mutations as well. This got me thinking that this might be a nodal event in podocytes that one has to worry about in terms of injury of the kidney filter, and perhaps more mechanistic work was needed to further understand this. Furthermore, work by last year's Homer Smith Award recipient, Professor Toshiro Fujita in Japan, had actually already told us that both in mouse models as well as in rat models, the activation of Rac1 was truly a nodal event. As shown in these examples from his work, his pioneering work essentially taught us that Rac1 activation is really a convergence node, a signaling node where multiple upstream pathways converge to activate several downstream pathways, all of them leading to the final event being podocyte injury. So all of this work, essentially both the human genetics as well as this excellent translational work done by Professor Fujita's group, was teaching us that we must care about the activation of Rac1 as a nodal event in podocytes. Now, this was very interesting to me because it drew in my mind a connection to another story. And that was, of course, pioneering work done by a friend and colleague in the field who I miss very dearly. We lost her way too young, Michelle Wynn, who had this pioneering study published in Science in 2005. It is fair for me to say that reading this study is really what got me singularly interested in podocytes and FSGS. And the reason for that is that I had devoted several years prior to that in doing a PhD focused on TRP channels. At the time, we thought that TRP channels were highly relevant to the brain and had no recognition of the fact that TRP channels would actually end up being relevant to podocytes and the kidney. And yet Michelle taught us that. Over the years, I developed a friendship with her. We exchanged ideas, and it was really valuable to have her as a person to talk to about our mutual interest in TRP channels. And as I said, we lost her way too young, but this work on TRPC6 and introducing the concept that TRP channels are relevant to the podocyte was actually instrumental in my thinking, my early thinking, as I started to develop this project. So on the one hand, we had Rac1, and on the other hand, we had TRP channels. And those two things were very connected in my brain because of this. This is from my own work as a graduate student, where we actually recognized that the activation of Rac1 in many different cell types, both in brain cells as well as in, turns out, podocytes, the activation of Rac1 is required for the activation of the TRPC5 ion channels. Not TRPC6, as Michelle was initially suggesting, but another channel, TRPC5. That got me thinking that there may be multiple stories in the podocytes that have to do with TRP channels, and I really wanted to uncover what was going on with TRPC6 and what was going on with TRPC5, specifically as it related to Rac1. And so we went on to do that. This is work that was done in the early days in my laboratory, and these are some of the very talented people who worked with me at the time: Thomas Schaldecker, Suk-Young Kim, and Konstantin Tarabanis, all of whom have moved on to medical school. Suk-Young is MSTP-funded MD-PhD student currently. I'm always eternally thankful for the work they have done in the lab, because it helped us sort out early on what are the distinct roles of TRPC5 and TRPC6 in podocytes. For TRPC6, it was no surprise. It is clearly relevant to maintaining the actin cytoskeleton in podocytes. But what we found was that TRPC5 was connected to Rac1 to develop an injury pathway, a pathway in which Rac1 inserts TRPC5 channels that are held in vesicles into the plasma membrane of podocytes, as shown here. Further, TRPC5 being activated at the plasma membrane allows calcium to come into cells, which further activates Rac1 through a calcineurin-dependent mechanism that we discovered, and that is partially shown here. Ultimately, this leads to a destruction of the podocyte cytoskeleton and podocyte injury, and that was how we were beginning to uncover a molecular pathway linking Rac1 to TRPC5 that was of potential injury to podocytes. To probe the mechanisms mediated by TRPC5 and TRPC6 and to parse their distinct roles in podocytes, we needed to study a model in which we could observe the progression of FSGS over time, very similar to patients. And so for this, we collaborated with Sigrid Hoffman at the University of Heidelberg, and we made use of an excellent rat model she had built, which expresses the angiotensin type 1 receptor specifically in the podocytes. TRP channels are receptor-operated channels activated by the AT1R, and so we could study the glomeruli of this rat model in order to parse the distinct roles of TRPC5 and TRPC6 in podocytes as the disease progresses. And in fact, what you appreciate here is the progressive proteinuria in this rat model that we established. And then through excellent efforts by Yiming Zhu, who was a postdoctoral candidate in the lab at the time and is now a professor in his own right, we were able to perform detailed electrophysiology studies looking at glomeruli isolated from this rat model and being able to land with a recording electrode on the podocytes in these glomeruli so that we could perform single channel recordings looking specifically for TRPC5 channel activity as compared to TRPC6 channel activity in these podocytes. And as you can appreciate here, what Yiming found is that over the course of time, in animals that have more advanced disease, there's an ever-escalating activity delivered by the TRPC5 channels, not by TRPC6 channels in this case, in these glomeruli. So therefore, TRPC5 activity was found to escalate and correlate with FSGS disease progression, which was really quite fascinating. This was work that was done in collaboration with another talented scientist in the lab, Phil Castonguay, who is now taking the biotech world by storm. I'm equally proud of both of them. TRPC6 is important, and it is there to perform a fundamental homeostatic role. It is there to preserve the actin cytoskeleton of podocytes. But when upstream pathways are activated that result in the activation of Rac1, TRPC5 is turned on, and that escalates together with proteinuria, indicating that TRPC5 is actually the driver of disease in most cases of FSGS where Rac1 is activated, but leaving, of course, room for the understanding that in the case of gain-of-function mutations in TRPC6, which Michelle Wynn had so elegantly demonstrated, that would be the time for a TRPC6 inhibitor to block the proteinuria. In most other cases where Rac1 is activated, where the cytoskeleton is destroyed, it is really the role of TRPC5 that we began to appreciate in this rat model. And again, it took detailed electrophysiology experiments in order to get there, which really points to the fact that no study of ion channels should be devoid of electrophysiology, as that is not really appropriate. There's no other appropriate measure looking at ion channel function. And so this was extremely informative, and it helped us to parse the roles of these two distinct ion channels in this complex system in the podocyte. With the additional help of Corey Hopkins, a medicinal chemist at the University of Nebraska, we were actually able to take it one step further. We built a specific TRPC5 inhibitor. Prior inhibitors were not clean for TRPC5 alone, but with help of Corey, we were able to do that. And now using this TRPC5, this specific TRPC5 inhibitor, we were able to go back to the rat models and demonstrate that administration of the TRPC5 inhibitor is able to suppress proteinuria. Again, the gold standard in the field, doing electrophysiology from isolated glomeruli from these rats, allowed us to demonstrate that AC1903 specifically blocks TRPC5 current in the podocytes of these glomeruli. And so in summary, in this very elegant graphic that was in reference to our paper published in Science a few years ago, it really nicely summarizes what the healthy podocyte looks like, where TRPC5 is sequestered in vesicles. It is not active, and the podocytes are happily able to sit on the basement membrane and mediate the filtration barrier, as compared to the case in which upstream receptors such as the AT1R are able to activate Rac1, bring TRPC5 ion channels into the plasma membrane, have those activated and allow calcium to come in, which further exacerbates the activation of Rac1, and on the cycle goes, this positive feedback loop that ultimately results in cytoskeletal changes that result in the detachment of podocytes from the plasma membrane, from their basement membrane, and their loss in the urine. And this is also summarized here, also highlighting the point that calcineurin is actually activated by the calcium coming in through TRPC5 in order to mediate the Rac1 activation. And this is in fact famously the target of cyclosporine, which we all know is a drug that we use in many of our FSGS patients. And so what I would like to propose is that the understanding, the detailed mechanisms that we understand as part of this pathway allow us to build now TRPC5 inhibitors that may be able to provide the same benefits as cyclosporine in those patients in whom cyclosporine works, without actually any of the side effects of cyclosporine, which of course we all know as clinicians and would prefer to avoid. And so this is a wonderful opportunity to build some new therapeutics in an area where there's a great unmet need for new treatments for this disease. And this is indeed what has happened. So genes to mechanisms to medicines has really played out nicely so far in this case, starting with a realization of Rac1 activating mutations in pioneering work done by the geneticists in our field, has led us to an understanding of a disease pathway, a Rac1 TRPC5 disease pathway in podocytes. And ultimately, I'm glad to say now we have a TRPC5 inhibitor that is currently in phase 2 studies in patients, really fulfilling this arc of discovery and providing an exciting opportunity for new treatments for our patients. And of course, I'm hoping very much that we will be able to successfully complete these clinical trials and hopefully demonstrate a benefit for our patients, because again, the unmet need is great. So having fulfilled this arc of discovery at the level of the glomerulus, we wanted to know whether this would be possible again with a new set of kidney diseases. And so this time we shifted our focus to tubular epithelial cell injury, a new category of diseases, and we looked to find genetically defined causes that may be able to provide insight into specific molecular pathways of interest. Specifically, we became interested in MUC1 kidney disease, otherwise known as autosomal dominant tubular interstitial kidney disease, MUC1. The causal mutation, I will tell you the story in a moment, was identified at the Broad several years ago. The Broad Institute is where I do a lot of my scientific work. And so then the question became, can we identify the molecular disease pathways in the relevant cell types, in this case tubular epithelial cells, and could that molecular understanding provide insights into a targeted therapy? Backing up for a moment, the story of MUC1 kidney disease is quite interesting. Known as autosomal dominant tubular interstitial kidney disease, it's autosomal dominant in its inheritance, but it is so rare and presents with such a bland urine that even most nephrologists may not have seen a patient with this disease or may not know that they have seen a patient with this disease. There are no treatment options other than dialysis or transplantation. And for a very long time, the haplotype was known. In fact, it was discovered by the Deltas group at the University of Cyprus. However, there was a long search for the gene that would be causal for this disease, which ultimately culminated in this 20-year challenge being resolved by perhaps arguably two of the world's greatest geneticists, that would be Eric Lander and Mark Daly, who several years ago at the Broad set out to finally solve this puzzle and be able to identify the causal mutation in these patients. And of course, what they found makes now sense in retrospect for why it was so difficult. It was literally like looking for the proverbial needle in a haystack. The mutation was hiding in perhaps one of the darkest corners of the human genome. And that is, of course, the variable tandem repeat region of the mucin-1 gene that was in this area. This is a single cytosine insertion that causes a frame shift and an early stop codon that results in a protein that bears really very little resemblance to the wild-type normal version of mucin-1, which of course has a transmembrane domain. It is a well-known glycoprotein that is presented at the apical surface of many epithelial cells. In this case, this frame-shifted mutant protein is comprised still of the signal peptide and a small part of the wild-type VNTR, but then has a large portion of frame-shifted VNTR, and again, an early stop codon that makes it impossible for it to have a transmembrane domain. It can no longer localize normally as the wild-type version would. Again, this cytosine insertion in a highly repetitive region was very hard to find. Next-gen sequencing methods do not work in this area, and so it really took heroic efforts, including traditional Sanger sequencing, in order to identify this mutation. Once it was done, in collaboration with Eric Lander, we began to ask questions about, well, what does this mutation do to tubular epithelial cells? How does this disease arise? And this is where my group became involved in answering some of these fundamental questions. A very talented postdoc in the lab, who is now an assistant professor at the Bar Ilan University in Israel, again, incredibly proud of Moran's work, Moran Vela-Levitt, took on the challenge. And together with other team members, decided that one of the fundamental early pieces to the puzzle would be developing a mouse model that hopefully recapitulates the disease. This is what is shown here. We developed MUC1-FS knock-in mice. We basically deleted one of the alleles of the MUC1 gene in the mouse and replaced it with the human mutant allele. We also made a mouse that had the replacement of the human wild-type allele as a control. And what we could see was that in the case of the knock-in of the mutant allele, we could easily detect over the course of a significant period of time, mimicking the human disease, which develops over decades, we were able to find the same dilated tubular appearance in these kidneys as seen in patients' biopsies, patients who have MUC1 kidney disease. This was very gratifying because it told us that we had an initial mouse model in which we could study the disease, and it really empowered our future studies. Looking a little bit more closely, here is a specific antibody that was raised to the frame-shifted version of the protein so we could tell it apart from the wild-type version. You can easily see that the green mutant protein accumulates in the same dilated tubules where the problem is in this mouse. And so we could begin to see that the accumulation of the mutant protein might in fact be part of the problem. We looked further and over time, and what we found is that the accumulation of the mutant protein over time, in fact, kills tubular epithelial cells. This is how the disease occurs. So this is a toxic gain-of-function mutation that results in MUC1 kidney disease being now classified as a toxic proteinopathy. In order to look at the human relevance of these findings beyond the mouse model, we worked with human kidney organoids, and this was work that was pioneered by a talented MD PhD student in the lab, Irini Sidhom, as well as our stem cell biologist, Mahesh Amani. They were both responsible for developing previously established protocols from very famous groups in the field like Melissa Little's group and Joe Bonventry's group. We were able to adapt these protocols and use them in this case to study mechanisms of disease and the development of therapeutics. And so in this particular case, I am showing you a kidney tubule of an organoid of a patient with MUC1 kidney disease. So this was basically pluripotent stem cells that were derived from blood taken from this patient with their consent. We took these pluripotent stem cells and developed organoids. And then staining these organoids, again, for the mutant version of the protein, you can see that it accumulates intracellularly, as we saw before in the mouse model. And the wild-type version of the protein, I remind you, this is autosomal dominant disease, so these patients also have a normal allele, MUC1 allele. The wild-type version of the protein, therefore, is appropriately apically localized in this tubule. Again, this was an excellent tool that would allow us to look at the human relevance of a lot of these pathways and molecular mechanisms that we were uncovering. Finally, we were able to develop an immortalized tubular epithelial cell line from a patient with MUC1 kidney disease. And again, you can appreciate that the mutant version of the protein is intracellularly accumulated, while the wild-type version of the protein is apically localized as expected. Armed with all these tools, we were now poised to do more mechanistic work and possibly begin to carve a path toward therapeutics. And in fact, this is what we did. Through collaboration between Mohan and Masha Alimova, a senior scientist in the lab, we did a high-content screen, screening several thousand compounds. I don't have time for the details today, but suffice it to say, we were looking for a compound that could do exactly what is shown here. We were looking for a compound that would leave the wild-type version of the protein intact while removing the mutant version of the protein. And we found such a compound. We were very lucky indeed to find this compound called BRD4780, a small molecule. However, very potent, with an IC50 of 150 nanomolar, it was able to very nicely and dose-responsively reduce the mutant version of the protein while leaving the wild-type protein alone and having no observable toxicity to the tubular epithelial cells, suggesting that this was a strategy that could provide insights for future therapeutics in this area. We wanted, of course, to test this small molecule in our mouse model, and this is what is shown here. This is a wild-type control mouse. This is the knock-in mutant mouse. As you can see, there's an abundance of mutant green frame-shifted protein, misfolded protein accumulating. But 1 week of treatment of a littermate mouse of this mouse with the BRD4780 compound resulted in a very significant reduction in the accumulated misfolded protein, lending further support to the idea that we were on the right track. Of course, we wanted to test the same strategy in human kidney organoids to, again, augment the relevance of this for patients. And we were very gratified to find that in 3 patients with the disease, we had significant accumulation, again, in the tubules. The wild-type version of the protein, again, is apically localized. And here's a control, which is a sibling of one of these patients. We had a matched sibling for each of these patients as a control. As you can see, there's no green protein accumulating here. These patients do not carry the mutation, the sibling controls. But after 3 days treatment with the BRD4780, we were actually able to very significantly reduce the accumulated MUC1-FS protein in the organoids of these patients, as shown down here, making them essentially indistinguishable from the control, the normal control, suggesting again that the BRD4780 compound may in fact have the ability to clear this misfolded protein specifically in patient tubules as well. So how does it work? How does BRD4780 do what it does. With a series of graphics, I would like to summarize a lot of detailed cell biological and biochemical work that we are doing and that is ongoing in the lab. But essentially, here's the normal condition. This is a tubular epithelial cell. This is the nucleus with normal DNA. In the case of the normal protein, the MUC1 is made in the endoplasmic reticulum of the cell. It further traverses through the secretory pathway to ultimately find itself inserted appropriately into the plasma membrane. That's what normal mucin 1 does. However, in the case of MUC1 kidney disease, we have one allele that carries the mutation. In this case, what happens to the MUC1-FS protein is that it begins to be made in the endoplasmic reticulum where it appears to initially accumulate. And then, this is the misfolded protein, and then the cell is trying to cope with this misfolded protein that makes no sense. This is a protein that shouldn't be there, so it's trying to cope. And one of the mechanisms by which it does so is to upregulate a protein called TMED9. It is otherwise known as a cargo receptor. So it sees MUC1-FS as cargo, it binds that cargo, and it begins to transport it toward the Golgi apparatus. Once it reaches the Golgi, quality control measures are kicking in, and they say, wait a minute, this is a misfolded protein. It doesn't really belong on the plasma membrane. It lacks a transmembrane domain. What can we do with this protein? Well, there is a little bit of degradation that can occur in the lysosome, otherwise known as the trash can of the cell. But really what mostly happens is that this TMED9 cargo receptor remains bound to its cargo and accumulates in this post-Golgi COP1 compartment where it just begins to ever so accumulate over time. And so this is the mechanism by which the cell is initially trying to cope with the misfolded protein and over time contributes to its further accumulation, basically in the wrong place. What the BRD molecule can do, though, is accentuate the cell's endogenous ability to take some of these misfolded proteins and degrade them in the lysosome. And it does so as described here. In this case, the vesicles are formed, but in the presence of the drug, more of the TMED9 and its cargo end up in the lysosome, much more of it, such that the cell can efficiently clear the accumulated both TMED9 and MUC1-FS, leaving none behind. This is the reason that BRD4780 is so efficient at clearing all of that misfolded protein, both in organoids, in cells, as well as in the mouse model. Armed with this understanding, and again summarizing here the fact that we have clearance of this mutant protein in several systems, in cells, in mice, as well as in human organoids, several people in the lab, talented MD PhD student Alyssa Campbell, and two postdocs, Silvana Bazua and Valeria Padovano, are now working on further mechanistic details to more fully understand the role of TMED9 in tubular epithelial cells and how this plays into binding of this misfolded protein cargo. Essentially, all in all, what we have uncovered here is really an exciting new mechanism for targeted protein degradation using an endogenous system that allows us to shuttle misfolded proteins into the lysosome. And there's so much more work to do to more fully understand and harness the potential of this pathway. Of course, if we're going to make our way to the clinic, we need to also think about the patients. This is a rare disease, and so with the help of several colleagues around the world, and they're shown here, Tony Blyer at Wake Forest, Greg Papagregoriou and Konstantinos Deltas in Cyprus, and also Stan Kmok at Charles University in Prague, we have collaborated with several other colleagues, many of you in the audience, to form the International ADTKD Patient Registry. And this is really a registry that over time has taken us from a handful of families, at best, that were initially described, to the discovery of the causal mutation and the development of a diagnostic test that is offered freely by the Broad, such that we can begin to accumulate more data and greater understanding of the natural history of this disease over time. And I'm very proud to say that as we speak today, this registry is about 1,800 patients strong, which really gives us much more opportunity to understand this disease more deeply and also develop the potential for clinical trials down the road. Instrumental to this effort is the ability to detect the disease and also how a potential drug would ameliorate the disease by appropriate biomarker work. And this is an area that is very active in the laboratory and is spearheaded by Julie Roignon, a postdoc in the lab, who's working very closely with other team members and with our collaborators in order to build an appropriate biomarker. So all in all, this work has led us to developing again an arc of discovery that starts with a causal mutation leading to misfolded proteins. We have understood the molecular underpinnings of this mechanism, a misfolded protein that is bound by TMED9 in the secretory pathway, and we have even uncovered a lead compound, BRD4780, that targets TMED9 and is able to remove the toxic protein in the kidney as well as other tissues. So armed with this understanding, we now have the opportunity to build new therapies for toxic proteinopathies, not only in the kidney for MUC1 and HUMOD, but potentially in the eye, the brain, and many other tissues. This is really an exciting time. And of course, this builds on Donald Seldin's incredibly transformative legacy for our field, as well as many other fields. He's considered the father of nephrology. He founded this society. He also founded UT Southwestern and became a major driving force for physician scientists all around this country. Many famous physician scientists who I very much admire at this time started their careers and owe their careers to Dr. Seldin. And it is more than appropriate that even during his lifetime, a bronze statue, a 7-foot bronze statue, was erected at UT Southwestern and dedicated in his honor. And this is actually Dr. Seldin in front of this statue, and it's so very appropriate for the truly transformative role that he had for our field. It has been the greatest honor and pleasure for me to receive this award in his name, as well as 2 years ago, the American Society of Clinical Investigation Seldin Smith Award for Pioneering Research, again, an award at the national level named after Dr. Seldin. I truly can't think of a greater honor for a nephrologist, physician scientist like myself, and I am very grateful for this recognition. Of course, everything that I described to you could not be done without the hard work of members of the team, our lab alumni, and a very elite team of collaborators all around the world who help us gain insights and make new discoveries. I want to acknowledge every one of the people named on this slide, but of course I cannot name them all by name. I just definitely want to acknowledge Eric Lander, who has given me the opportunity to develop my scientific career at the Broad Institute. Amin Arnoux, who gave me my first job at the Mass General, and Joe Bonventry, who gave me my second job at Brigham and Women's Hospital. I am truly indebted to them for their support. Betsy Nabel, the president of the Brigham, has been a supporter of my career, and I'm truly grateful to her. I'm most grateful to a whole host of collaborators all around the world in different contexts who have been instrumental in our lab being able to make the contributions that we have made. And I am truly very grateful to all of our patients, without whom we couldn't make the progress that we made. I think all along my talk today, you have been able to see how often we've been in a position to partner with patients, and that has been instrumental in us being able to make adequate progress. And so I'm truly grateful to patients and their families for being involved and continuing to be involved in all of our efforts. This is some of the pictures from our laboratory as it has grown over the years. It is a wonderful team, supportive, generous, insightful, talented, and it's really the greatest honor and pleasure of my professional career to work with them. If I think about my beginnings, though, I must acknowledge David Clapham. I was an MD PhD student in the Harvard-MIT HST program, Health Sciences and Technology program. And it's been tremendous to train in that environment. But most of all, I'd like to credit David, because he is the person who really taught me how to think as a scientist, how to write as a scientist. He has been incredibly important to my career, and I couldn't be more grateful for the opportunity to work with him several years ago.
But of course, if I would go way back, my first teachers were actually my parents, Dr. Dimitris Grekas and Dr. Harula Kaleku Greka.
I grew up in Thessaloniki, Greece, as shown here. And my parents never hesitated to remind us that we were very near the birthplace of one of the greatest minds of all time, scientific minds as well. And that's, of course, Aristotle. Truly, his birthplace was just a few miles down the street from where I was born. And my parents instilled in me curiosity, strive for excellence. And in many ways, all of the values that I gained from them, my very first teachers, are encapsulated in this wonderful quote by Aristotle. Excellence is never an accident. It is always the result of high intention, sincere effort, and intelligent execution. It represents the wise choice of many alternatives. Choice, not chance, determines your destiny. I am forever grateful to my parents, who are still my moon and sun, for everything they have given me over the years. I'm truly grateful. But of course, this crew is really, in my heart of hearts, what's most important. This is my family. I am incredibly lucky to be married to my soulmate, Peter. He will probably cringe hearing me say this, but he also knows that it's true. And our incredible kids, this is Dimitri, Gilly, Ella, and Constantine, also shown here. It is really, truly the case that with all of my heart I see that while everything that I've spoken about today is extremely important to me, at the end of the day, it's this crew and the support and love that they give me that makes everything else possible. And for that, I'm truly grateful to my family for everything they do for me every day. And with that, I'd like to thank you all for your attention. And again, it is a tremendous honor to be able to accept the Donald Seldin Award today. Thank you.
Hello, I'm John Klein, and I am co-chair with Linda Freed of the Education Committee for this Kidney Week. Thank you all for joining us for this third day of Kidney Week. I hope that you've enjoyed the educational content, the exhibits, and the networking opportunities. Today's state-of-the-art lecture will be delivered by human geneticist and genomic medicine researcher Heidi Rehm on the topic of global efforts to curate the genome. Dr. Rehm is medical director of the Clinical Research Sequencing Platform and an institute member at the Broad Institute. She's also chief genomics officer in the Department of Medicine at Massachusetts General Hospital and professor of pathology at Mass General, Brigham and Women's Hospital, and the Harvard Medical School. She's a leader in defining standards for the interpretation of sequence variants and is principal investigator for a major NIH-funded effort called ClinGen, which provides free, publicly accessible resources to support the interpretation of genes and variants. Dr. Rehm also co-leads the Broad Center for Mendelian Genomics, which is focused on discovering novel rare disease genes. She's a pioneer in open science and data sharing and is working to extend these approaches through her role on the steering committee of the Global Alliance for Genomics and Health. She's also co-investigator of the BabySeq project, exploring the clinical use of genomic sequencing as an adjunct to newborn screening. In 2001, Dr. Rehm began building the Laboratory for Molecular Medicine within the Harvard Partners Center for Genetics and Genomics. The laboratory focuses on the rapid translation of new genetic discoveries into clinical tests and brings novel technologies and software systems into clinical use. The laboratory, which she directed until 2018, has been a leader in translational medicine and offers exome and genome sequencing services. Dr. Rehm is involved in defining standards for the use of next-gen sequencing in clinical diagnostics and the interpretation of sequence variants through her committee roles at the American College of Medical Genetics and Genomics. Among her many honors, she has received the Brigham and Women's Hospital Physician Recognition Award for Clinical Innovation and was named by Boston Business Journals as one of 40 under 40 for civic leadership. She was a member of teams that won the 2012 Clarity Challenge run by Boston Children's Hospital and the 2013 Editor's Prize for the Gene Insight software system. Now I'll turn it over to Dr. Rehm, and please give her a very warm welcome.
Hi, my name is Heidi Rehm, and it's a pleasure to be here at the ASN conference, even if virtually. And I'm going to talk today about our global efforts to curate the genome. So I like to start my talks off with a real story that helps contextualize our work. Over 6 years ago, my lab detected a PTPN11 variant in a fetus with increased nuchal translucency seen on fetal ultrasound. And that variant had been reported in an Ashkenazi Jewish individual with Noonan syndrome and was reported as pathogenic by a very well-respected lab. Now, in the context of interpreting this variant in my clinical lab, I had tried to get access to a research data set of Ashkenazi Jewish individuals to try to determine the allele frequency, but was denied access to that data set. They just weren't ready to share. So we used the evidence we had, we reported the variant as likely pathogenic, and the couple terminated the pregnancy. Now, several years later, we found out that that variant was in fact at high allele frequency in the Ashkenazi Jewish population. In fact, from that very data set I was denied access to, and that data showed the variant was in fact benign. So this outcome really could have been avoided through data sharing. The good news is that now we have the ClinVar database working in partnership with ClinGen. We have the Nomad database at the Broad Institute, started by Daniel MacArthur. And these resources have dramatically changed how it is we are able to interpret variation and give results back to patients. And in fact, several years later, a very different story based on a very similar situation happened. In this case, a couple contacted me having had prenatal genetic testing that was prompted by, again, an increased nuchal translucency measurement. That testing revealed a variant of uncertain significance, and the couple was savvy. They discovered the ClinVar database. They found that my lab had submitted an interpretation on that variant to ClinVar and contacted me just asking, do you have any other information about this variant? They were concerned about their pregnancy. So I dug up the case that we identified this variant in. I also reached out to GeneDx and had also submitted a variant interpretation. And we aggregated the data together and were able to reclassify that variant as likely benign. And the couple continued their pregnancy and had a healthy baby boy that very next fall. So this was a very different outcome, largely based upon having robust data sharing. And those variants are now reclassified as likely benign in the ClinVar database based on that work. So I would argue that for the best outcomes for patients, we need to enable data sharing on a global scale and critically evaluate the evidence as a collective community. I'm now involved in running the Nomad database along with Mark Daly, and we continue to add more exomes and genomes. The V4 release in next spring will have approximately 500,000 exome and genomes, or the aggregate data from that resource that will be publicly available. And this is continuing to assist all of us in interpreting genomic variation across our genomes. So now I'd like to talk a little more in depth around what ClinGen is actually doing. So our goal is really to create an authoritative central resource that defines the clinical relevance of both genes and variants for use in medicine and research. And we start with aggregating genomic data from all stakeholders, patients, clinicians, labs, researchers, and then we curate that data based on key questions. Is this gene-disease relationship valid? Is this variant pathogenic? Is this region in the genome dosage sensitive? And is this information actionable for a patient? We curate that knowledge and then we disseminate it back to those same stakeholders to use in improving patient care. This program was started in 2013 and is primarily funded through 3 core U41 genomic resource grants supported by NIH. And we also have smaller disease-focused grants being funded, and in fact, a whole nother round of those grants will be funded shortly. So this project involves many people around the world. In fact, we have over 1,400 researchers and clinicians from 35 countries that participate in our working groups and expert panels. And in order to manage that, we have phone calls at every hour of the day, depending on where you are in the world, but we make it work, and it's a lot of fun to work together as a community. Now I get asked the question, what's the difference between ClinGen and ClinVar all of the time? So I came up with this little slide to help describe the difference. I think of ClinGen as a consortium of people sharing data, developing standards, curating knowledge, whereas ClinVar is a variant database. It's actually maintained by NCBI, where everyone submits their variants so that we can share them with the collective community. We also maintain clinicalgenome.org, which is where our gene-level database is for our curated gene-level knowledge. Now, if we get into the details of curating, really before we can define a variant's role in disease, we first need to assess the gene-level evidence. And that's a big area of ClinGen's work, is gene disease validity curation. Now, to do that, we've developed a semi-quantitative framework to classify the strength of evidence for the role of genes in disease. And to do that, we use both genetic evidence that we score, case level data, segregation, case control analyses, as well as experimental evidence, expression, model organisms, rescue studies, etc. And all of that curated knowledge and evidence leads us to classify each gene-disease relationship that's been reported into either definitive, strong, moderate, limited, no evidence reported, or conflicting evidence, either disputing or refuting the relationship. We curate these genes using expert panels that have disease-specific expertise in different clinical domains. So we organize them by broad clinical domains like cardiovascular disease, neurological diseases, and then we form these expert panels that either do gene curation or variant curation, or sometimes start with one and move to the other in sub areas of these broader domains. And on our website is a list of all the different clinical domain working groups and the expert panels they support. On our website is then the gene level results of our curation. So this is an example of the CHEK2 gene. Now this gene has actually been implicated in 3 different conditions: breast cancer, ovarian cancer, and colon cancer. And what you can see here is that the breast cancer has been curated as definitive, but the colon cancer was limited and the ovarian cancer disputed. You'll also see we have curations for actionability as well as dosage sensitivity, suggesting there is sufficient evidence for haploinsufficiency if that gene is deleted, but no evidence for triplosensitivity if there's an extra copy. So we've curated genes across many different disease areas. To date, we've curated over 1,085 genes. And the results of that curation suggest that to date, about 65% of genes score in the definitive to moderate range, meaning there's good evidence or pretty good evidence that that gene has a role in disease. However, about 35% of those curations result in insufficient evidence, meaning that reported relationship really was not valid from the start. And we've done this work across many different disease areas that I'm showing here. Most have similar ranges. We have one, probably the worst example, is Brugada syndrome, where of the 21 genes implicated in that disease, only one of them was considered valid. The other 20 were disputed by that expert panel. So that's a sense of how we're doing this gene curation. Now, how is this information used? Well, it certainly helps researchers point to areas where they need to do more research and generate more data. But also in the context of clinical testing, we can use these categories to help inform which genes should go in what types of tests. For example, I would argue that predictive tests for healthy individuals really should rely on only genes that we really understand well, the strong and definitive relationships. For diagnostic panels, testing patients who are ill with those conditions, I think it's feasible to include the moderate level genes, often those clinical cases that we start testing in clinical care, often build the additional evidence that bring it up to definitive and strong. However, all of those genes that have been curated as limited or below really have insufficient evidence and shouldn't be in a diagnostic panel or predictive test, and should be relegated to exploratory analyses, either clinical exome and genome sequencing, where we are looking at unknown genes, or research studies. So how have the laboratories been using this information? Well, this is an example of Invitae, one of the largest genetic testing laboratories, and when the data came out from the Brugada syndrome curation, they changed their panel. It now has only one gene based on the results of that curation. They do allow clinicians to add on what they call preliminary evidence genes, and I think that's a good balance. The default is the one gene, but they do allow clinicians, if they want to explore these less understood genes, they can. Now, ClinGen's not the only group that's been curating gene disease knowledge. There are other resources that are public, like OMIM, other clinical labs that have curated gene disease relationships. So a number of years ago, we got this group together to clarify overlap and what groups were curating and what their approaches were, as well as develop consistent terminology, really with the goal of then sharing all of our data. So we went through a a Delphi survey process to harmonize on our terms. What you can see here is the initial set of terms across the public resources that were available at the time, and you can see they were using all different terms and color systems, in fact, opposite color systems for what those colors meant. So through this Delphi survey process, we were able to bring everyone into a single harmonized set of terminologies. It's very similar to the ClinGen terms, but we did add animal model only. These are genes that have been validly implicated in animals, but not yet in humans. So we are working to ingest the data from all of these different resources. Right now it's in progress, and we'll be launching in December a website that brings all of this data together. This is just a screenshot of the mock-up of what this will look like, where you can search for a disease area and all the genes will come up in that area. If we drive a little deeper, this is a more detailed mock-up when you open up one of these genes, where you can see the different numbers of curations in these different classifications and the sources of those classifications and for what diseases those genes were curated. And that allows one to quickly not only see ClinGen's results, but see all of the results together at once, including sources that have not been publicly available to date from many of the clinical laboratories. So we're very excited for this resource to launch. It should be live within the next 2 months. So now we're going to move on to variant curation, and that's a much more challenging area to tackle because we have over 750 million variants. We can't possibly expert curate them all. So how do we address this? So we take a more four-pronged approach, as I like to call it, and the first of those things is to develop common standards. That enables the entire community that's curating variants in their ongoing work to apply consistent standards to improve the work that we all do together. So to start that, we worked with the American College of Medical Genetics and Genomics and the Association for Molecular Pathology to come up with a better framework for how we define individual types of evidence and how we combine that evidence and how we classify those variants into a consistent set of classifications which span pathogenic, likely pathogenic, uncertain significance, likely benign, and benign. And these standards that we released in 2015 have been adopted worldwide and have been very helpful in improving the consistency of how we interpret variation. However, many of us have recognized that there's still subjectivity to applying these criteria. So ClinGen formed its own working group to tackle this and really build off of the ACMG AMP guidelines. Our working group is chaired by Les Biesecker and Steven Harrison, and the goal is to really refine and evolve these guidelines as they've been tested and deployed by the community, and we've identified areas of more variation. The group also reviews and harmonizes the gene and disease specific specifications of those rules that our expert panels are coming up with for their individual genes or sets of genes. And we're also gradually moving this framework to a more quantitative approach using a Bayesian underlying framework. All of the work that our SVI working group does is then posted on the ClinGen website. Some of that work is in the form of publications, either generally from our group or from the disease-specific expert panels, or if it's not published, it may be in white papers that we post online. And that's where you can go to get that information to help with your curation approaches. The next step we take is really supporting public sharing of variant interpretations. This creates transparency and it crowdsources the work. So to date, we have worked in partnership with ClinVar to support submissions from places all around the world. There are 1.3 million submissions from 846,000 variants from 1,716 submitters from 78 countries. So just a wealth of information that's coming in from all around the world. Just a great example of data sharing. Interestingly, 84% of the data in ClinVar is actually from the top 20 submitters, and that was in part intentional. We reached out to the largest clinical laboratories early on to work with them to encourage them to submit to ClinVar and really form the foundation of this database. And that includes 13 of which are commercial laboratories. So commercial labs voluntarily sharing their data along with the rest of the community has really just been a pleasure to see. Now, if we look at ClinVar, one of the benefits of all of us submitting to a single database, it allows us to compare our data. And although 3/4 of the variants we submit to ClinVar have only been seen by one laboratory, another 1/4 of it has been seen by multiple labs, and we can compare. Of that 1/4, so 15% of the total, those interpretations are concordant. Another 5%, they're similar, just a confidence conflict. 5% there differ between VUS, uncertain significance, and likely benign or benign. And then there's 1% of variance across the entire database that represent what we call medically significant conflicts. Over 9,000 of them to date that we've been working to tackle across all submitters. If we focus just on the 1-star submitters that share their evidence and have consistent methods, that's about 5,000 variants that we're working to resolve. So first off, we've been supporting inter-lab conflict resolution. So to do that, we bring labs together. In the first round of this, we had 4 labs that basically reassessed all of the variants and then figured out when they would agree on it and why they were differing in their interpretations. What we found from that is most of the time, the interpretation was simply out of date or hadn't been classified with current guidelines, and just re-looking at the variant quickly resolved things. However, a good percentage of those, about 1/3, were because of data that only one laboratory had in-house. Segregation data, de novo occurrence, that kind of data that the other lab didn't have access to, and by sharing that evidence, both labs were able to come to agreement. And so through that very rigorous approach across a small number of labs, we were able to resolve 87% of the differences between the interpretations. Now we moved on to a slightly higher throughput approach, first with a round of 41 labs and then subsequently 108 labs. And that really, we took an outlier approach where first we asked the lab that was discordant with the rest of the labs to reevaluate, or maybe the one that was the oldest. Through that approach, you could quickly resolve a lot of these differences with a lot less effort. It didn't resolve quite as many as when everybody reassessed, but it did resolve the majority of those differences. And in the right column here, what you're seeing is even when we don't even organize efforts to compare laboratories and try to work to resolve, already we're leading to unprompted resolution about 1/4 of the differences, simply because one lab can see the evidence that another lab has submitted and use that in their reassessment. The last approach, and as I mentioned, even when we try really hard, we don't always get all of the variants to complete agreement, and that's where our experts come in. And in this case, we make use of the star system in ClinVar. So 4 stars in ClinVar means it's a variant classified through a practice guideline. 3 stars is through one of the ClinGen approved expert panels. 2 stars means multiple labs have submitted and they all agree with each other. 1 star is a laboratory that shares its methods for variant classification and shares the evidence supporting their claims. And then no stars is if you're not sharing your evidence or your method of classification. So each of those categories overrides the other in terms of what shows up as the aggregate classification in ClinVar. And so by using our expert panels, we can curate these variants through expert consensus, and then that overrides all of the individual laboratory submissions. And this is how we gradually sort of clean up the ClinVar database over time. I should also note that ClinGen worked hard over the course of a year to get FDA recognized database status on its submissions. That was a rigorous approach that we had to take to define our methodologies and ensure appropriate training and documentation and validation of systems. But now all of the ClinGen expert panels have this additional tag in ClinVar. And each of our expert panels goes through a rigorous process of defining their work group members. They have to span multiple institutions, define conflicts of interest and track those. Then they move on to developing their ACMG AMP specifications for the genes in scope. Then they pilot those rules with known variants. They may refine the rules over time. And then they must be approved through our SVI working group to ensure those rules that they've developed are not are harmonized across different groups and are taking appropriate methodologies. Once they've been approved, they can then move on to routine curation and submission to ClinVar. And the work now of the ClinGen expert panels has led to over 12,000 variants that have been expert classified. And in many cases, those have overwritten variants that have been discordant in ClinVar, as shown here. All of this data and the detailed evidence that supports our work is both in ClinVar, and then links go back to our evidence repository shown here, where you can see all the detailed rationale and evidence that supported the ClinGen curations. Now, we don't just focus on resolving discrepancies. Sometimes the variants everyone agrees are uncertain significance, but how can we move them into a state of more certainty? And so in doing that, we bring our clinical labs together and our research partners, and we bring and aggregate evidence together, and sometimes that allows us to move variants from uncertain significance up to likely pathogenic or down to benign. And this is just an example of several variants of the cardiomyopathy expert panel was able to move up to likely pathogenic by aggregating evidence together. And now I want to turn to our specific work in the area of kidney disease, since it is the ASN conference. We formed a clinical domain working group that's co-chaired by Andrew Mallett and Matt Sampson and coordinated by Tam Snedden. And you may have heard Andrew Mallett's talk yesterday, where he talked a bit about this as well. So this clinical domain working group has formed multiple gene curation and variant curation expert panels. We have 80 members from 36 institutions across multiple continents that are participating in this effort. We started with a GCEP in cystic and ciliopathy disorders that's co-chaired by Anna Greka, Peter Harris, and York Pei, and you can see the membership here on the right side. That group has formed both a gene curation expert panel, and will soon be starting our variant curation activities in that area as well. And then we'll follow that by a GCEP doing glomerular disorders, and then complement disorders, Cook and structural disorders, tubulopathy, and tubular disorders, really spanning all of the different major areas of kidney disease. So in terms of the first results hot off the press for this group, we started with a list of 121 genes in cystic and ciliopathy disorders that was drawn from a larger list of 760 kidney disease genes from multiple different clinical testing panels that the group brought together. This was work of Tam Snedden. And so far to date, we have curated about 11 different genes, and you can see here on the right that the curation is similar to the work I showed you earlier across other disease areas. Similarly, there are some genes that are moderate or definitive, others that have insufficient evidence, much like all of the disease areas we're finding. We're excited to continue this work and be able to better define all of the genes that have been reported as implicated in kidney disease. I should note down here the 6 pre-curations. A lot of the work starts out with trying to agree on which disease we're actually curating on for these genes, and how to name that disease, and whether the names that have been used in OMIM and Mondo ontology are correct. So you can imagine that generates lots of discussion already before we even get to the evidence for implicating the gene. But nonetheless, it's critical work that we bring the community together and create this resource that's critical for the work that we do. With that, I'll just invite anyone in the audience to actually get involved in ClinGen. You can go to our website, learn about the work we do and the ways to get involved, either as a volunteer, biocurator in one of our expert panels, or you can get, if you have disease-specific expertise and want to join one of our expert panels as a disease expert, particularly in the area of kidney disease, you're welcome to reach out and contact us, and we can bring you into the appropriate working group or expert panel. And with that, I'd just like to thank so many people that are involved in ClinGen. This slide gives an overview of all of our different work groups and the co-chairs of them, as well as the PIs and other members of our steering committee, the key coordinators, and our external scientific panel. But I do want to mostly highlight the over 1,423 people from around the world that participate in our working groups, volunteering their time to create a resource that's critical for all of us. And I really am ongoing excitement and appreciate all of that work. Thank you very much.